Photosynthesis is the process of converting light energy into a usable form ATP by the cells. Leaves, being the primary light-collecting organ of the plant, contains chloroplasts, a special organelles in the cell of a plant where photosynthesis occurs. Each chloroplast consists of the outer and the inner membrane. The inner membrane consists of layers of thylakoid stalks called granum. The grana are surrounded by a gel-like matrix called stroma. It is the thylakoid that converts light energy to chemical energy of ATP. Its thylakoid is a closed compartment. Space between its thylakoid is called lumen. Inside the thylakoid are groups of light-collecting molecules called photosystem 1 and 2. Along with them are proteins of electron carriers that serve as messenger from each photosystems, and also the presence of ATP synthase protein. When light energy strikes the plant, the photosystem 2 inside the thylakoid works as it absorbs the energy, causing the water molecules inside the cell to split into 2 hydrogen ions, 1 oxygen atom, and 2 electrons. The hydrogen ion stays in the thylakoid membrane while the electrons move into photosystems 2. With the continuous supply of light energy, the electrons are energized and moves into the first chamber of the electron carrier. Energized electrons are then moved to the second chamber, whereby the hydrogen ions outside the thylakoid are moved across the membrane. Thus, the presence of hydrogen ion in the membrane increases. Electrons then are transferred to the third chamber. Then they are moved to photosystem 1. As light energy strikes photosystem 1, electrons are energized and moves to the final carrier protein, wherein ADP plus is added to form NADPH. On the other hand, the high concentration of hydrogen ions inside the thylakoid membrane is kept balanced by diffusion through the membrane with the aid of carrier protein ATP synthase. In every active transport, ADP form of energy energize the carrier protein to pump out hydrogen ions, thus converting ADP to ATP to power other cell functions. From the light reaction stage of photosynthesis, energy is stored in the form of ATP and NADPH. This stored energy goes to the second stage of photosynthesis called the Calvin cycle. The Calvin cycle occurs in the stroma to produce sugar molecules. It begins with the input of carbon dioxide molecules, which are added to the 3 molecules of 5-carbon sugar molecules, or RuBP. These molecules, by way of chemical reaction, are rearranged, thereby producing 6 molecules of 3-PGA with 3 carbons each. Products such as ATP and NADPH from the light reaction are used to boost the energy of these 3 carbon molecules. First, high-energy phosphate group is added. 6 ATP molecules contributes to the phosphate group so that each of the 3 carbon molecules receives an additional phosphate group. In the next reaction, electrons are also added. Then 6 NADPH molecules are oxidized. Electrons from NADPH reduce the 6 carbon compounds, creating high-energy 6G3P, or glyceraldehyde 3-phosphate. G3P is a sugar which is the final product of the Calvin cycle. One of the three GP molecules represents the carbon dioxide molecule fix. The other 5 3GP molecules are then reshuffled to regenerate the original RuBP molecules. To summarize, Calvin cycle has used the energy ATP and NADPH from the light reaction to reduce 3 molecules of carbon dioxide and produce 1 molecule of 3GP. More so, 3 carbon dioxide molecules are then again fixed to form G3P, again with the same process. To make G3P, the Calvin cycle consumes 9 ATP molecules and 6 NADPH molecules, which are regenerated in the light reaction. G3P is the actual and final product of the Calvin cycle.